Welcome back to the deep dive. If you've ever stopped to consider how just bafflingly complex life is, you've probably heard someone say, well, DNA is the code of life. It's an easy analogy.
It is, but most people treat it as just that, an analogy. Our sources today push far beyond that. They argue that biological systems, genetics, metabolism, all of it are fundamentally systems of pure, precise computation.
So we're moving from messy chemistry to rigorous information science.
Exactly, to engineering.
And that's our mission for this deep dive, to explore the physical reality of biology as code. We're going to unpack three, well, pretty radical concepts. First, we're looking at research that maps the genetic code onto a mathematically elegant 6-bit binary system.
Then we'll dive into how DNA, this fundamental molecule, has been successfully turned into basically the ultimate digital hard drive. Dense, archival.
And finally, we'll look at how synthetic biologists are moving from just reading the code to actively programming it. We're talking formal languages and actual biomolecular logic gates.
Yeah, building biocomputers inside living cells. This convergence is, I think, arguably the most exciting field in science right now. It shows that the basic rules of computing, things like binary operations, are already built into the very fabric of life.
The ultimate engineering blueprint. Okay, let's unpack this, starting with the deepest code we know, the genetic code itself.
Right. So we all know the basics. 64 triplet codons specify the 20 standard amino acids. And for decades, scientists noticed patterns, redundancy, what they call degeneracy.
Where multiple codons code for the same amino acid.
Exactly. But recently, researchers, like the ones behind this NIMSARY system, they developed a totally new approach. A 6-bit binary representation of those 64 codons.
Okay, so a codon is a triplet of bases A, C, G, U. If you assign a unique 2-bit ID to each of those 4 bases, the codon just automatically becomes a 6-bit label. It sounds elegant, but why? Why go to all the trouble of imposing a binary system on a 4-base system?
Because it reveals a hierarchy that was always there, just hidden. The crucial insight is that these 6-bit labels aren't arbitrary at all. The ordering actually reflects the functional structure of the code, which is optimized for replication and translation.
How so?
Well, for instance, the binary assignments immediately separate the pyrimidine bases, that's U and C, from the purine bases, A and G. That chemical classification is instantly reflected in the bit string.
So nature's already kind of running on a quasi-binary structure, and the bits we assign just reveal that. But do those bits actually correlate with the output, with the protein's function?
Absolutely. This is where the engineering perspective gets validated. If this code is robust, the bits have to align with the properties of the amino acids. Size, charge, and most critically, hydropathy.
Hydropathy, that's the amino acid's affinity for water, right? Dictates how the protein folds.
Exactly. It's fundamental.
And what's the strongest link you found in the sources?
The analyses show that the most important correlation comes down to one single bit, the first bit of the second nucleotide in the codon.
Just one bit.
Just one. That single bit overwhelmingly dictates the hydropathy of the amino acid. So if that bit is a 0, you are far more likely to get a hydrophobic, a water-repelling amino acid. If it's a 1, you often get a charged hydrophilic one.
Wow, that's fascinating. That grouping suggests the code is optimized for error management. If a random mutation happens, you're more likely to swap an amino acid for one with a similar water affinity.
Which might just save the protein from catastrophic misfolding.
Exactly. The code is structurally robust.
And this binary view lets us move beyond just structural analysis and actually treat mutations as formal Boolean operations, as simple bit flips.
And this is where it gets really interesting. You've got 2 main types of natural mutation: transitions and transversions.
Right. A transition mutation, say U to C, it preserves the pyrimidine class. In binary, this just flips the second bit. It's a minimal information change.
And those are generally less damaging to the protein.
Much less damaging. But a transversion switching from a pyrimidine to a purine, like a U to an A that flips the first bit, or maybe both. It's a fundamental chemical switch.
Okay, so mathematically, simple chance would suggest that transversions should be twice as likely as transitions. You've got more ways for it to happen.
4 potential transversions versus only 2 transitions.
Nice.
Yet, when scientists actually look at real-world biology, they find the complete opposite.
Precisely. Transitions are observed to be about 3 times more common than transversions in nature. And that's a profound insight. It means the DNA replication and repair machinery has, well, a six-fold lower fidelity when it has to distinguish between bases in the same class.
So the system is actively, though chemically, optimized to avoid the most damaging bit flips.
It's an error-correcting code shaped by evolution.
Incredible.
Mhm.
So if the code of life is this structured, it immediately makes you wonder, can we use the physical DNA molecule itself as, a data store? And the answer, as proven by George Church's team back in 2012, is a definitive yes. We can hijack this molecule for massive, non-biological data storage.
Tell us about the sheer scale of that project. I know they didn't just encode Hello World or something.
Oh, far from it. They encoded an entire book that Church co-authored, Regenesis, along with 11 illustrations and even a functional JavaScript program.
A whole program.
A whole program. The total data set came to 5.27 megabits. Now, that might not sound like a lot today, but at the time, it was 600 times larger than anything that had been stored in DNA before. It was a massive proof of concept.
One of the key choices they made was about the encoding. DNA has 4 bases, so they could have used a 4-value logic system to increase density. But they didn't. They stuck to binary. Why?
Error minimization. It was all about reliability. They mapped A and C to represent 0 and G and T to represent 1.
Ah, so they built in redundancy.
Exactly. If a chemical reading error occurs and an A is mistaken for a C, the computer still reads a 0. It's a robust system, optimized for reliability over theoretical max density.
And the physical storage itself. I mean, DNA is famously tiny. What does this mean for archival density?
It's monumental. When you look at the density, it just completely dwarfs traditional storage. Hard drives are essentially two-dimensional. DNA is fundamentally three-dimensional.
You can pack all that information into a single droplet.
A droplet that would otherwise require a whole room of tape drives. It's estimated that a single gram of DNA can theoretically store hundreds of exabytes of data.
But wait, if they synthesize all this DNA into millions of tiny fragments on a glass chip, how do they ever read it back in the right order? That sounds like a complete nightmare.
That's the genius of the engineering. Each synthesized DNA fragment, each little piece of the puzzle, included a short, built-in digital address code.
So when they sequenced it all,
The computer didn't just read the data, it also read the address, which said, hey, I belong at line 4, block 7, position 12.
so the computer just reassembles the text perfectly. That's clever.
It's the only way to manage that kind of chaos. Okay, so we've established DNA is highly optimized code, and it's a brilliant hard drive. But to truly program biology, we need to move beyond just storage. We need formal engineering languages.
Right. We're moving from the passive role of observing the code to the active role of writing it.
Precisely. If synthetic biology is going to be a real engineering discipline, it needs abstract, defined languages, just like software engineering. And our sources highlight 2 specialized languages designed for this.
Let's start with LBS.
Okay, so LDS is the language for biochemical systems. It's developed for what we call systems biology. That's about modeling and understanding what already exists.
Like the yeast pheromone pathway or complex signaling networks.
Exactly. And before LBS, the modeling tools were kind of caught in a trade-off, right? Some were modular but so mathematically complex they were unmanageable. Others were intuitive but monolithic. You couldn't reuse code. LBS bridges that gap.
It sounds like it brings some much-needed structure to modeling, but what about designing something totally new?
That's where GEC comes in. The language for genetic engineering of cells. This is for synthetic biology. It's about design and construction. Instead of getting bogged down in the minute details of DNA, GEC lets the designer work at a high level using logical constraints.
So if I want a cell to light up green, only if input A and input B are present, I don't program every chemical step. I just specify the logic.
You specify the phenotypical characteristics, what the cell should do. And the GEC program automatically translates that abstract description into a physical DNA sequence. It pulls optimized parts, or biobricks, from a database to build it.
It's basically like moving from coding in assembly language to coding in Python.
That's a perfect analogy. And this modularity is key. LBS lets you reuse code to avoid redundancy. GEC lets you build incredibly complex systems, like the famous repressilator circuit.
Which brings us to the actual execution. We have the code, the hard drive, the programming languages. Now we need the processors. In computing, that's logic gates.
And in biocomputation, we synthesize biomolecular logic gates, the equivalent of AND or R and NOT gates that physically sense chemical inputs and execute a decision.
This technology seems to break down into 2 main camps: stuff you run in the test tube and stuff you build inside a living cell.
Yeah.
What's the major difference between them?
For anyone interested in the application, the biggest difference is speed. Cell-free systems in a test tube can execute logic in seconds or minutes.
Whereas cell-based devices are slow, they have to rely on gene expression, which can take hours, even days.
Right. But you get the stability of a living system. It's a trade-off.
So for these speedy cell-free systems, what's actually doing the logic?
Well, often nucleic acids themselves. You have enzyme-based systems like deoxy or ribozymes, which have been used to construct yes, not A and D, even X or R gates.
And I saw that scientists are even using the basic rules of DNA pairing for logic, without any enzymes.
Yeah, this stuff is ingenious. They exploit Watson-Crick base pairing dynamics. Techniques like branch migration and these newer seesaw gates use carefully designed DNA strands that compete to pair with each other. You can build A and D and OR logic through strand displacement alone.
The aptamer example was a perfect illustration of this.
It really was an aptamer-based A and D that required two distinct inputs, adenosine and thrombin, to both be present to get a fluorescence output. If only one input was there, nothing happened.
So the logic is literally baked into the DNA structure. What about using proteins for this?
That's a separate challenge. Researchers have used cocktails of enzymes like glucose oxidase and catalase that operate on inputs like glucose and hydrogen peroxide. By linking these reactions, they built 7 different logic gates.
And even a half adder, I saw.
And a half adder, yes.
Okay, for those of us who aren't electrical engineers, why is a half adder such a big deal?
A half adder is the simplest possible circuit that can perform binary addition. It takes 2 inputs and produces a sum and a carry bit. Building this biologically is a monumental step. It proves that complex arithmetic processing is achievable with biomolecules.
But the big challenge with these cell-free systems is noise and interference. Why does moving all this inside a cell help solve that?
Sequestration, compartmentalization. Inside the cell, things are naturally separated by membranes. This drastically increases the signal-to-noise ratio and reduces crosstalk between all the circuit parts.
Let's go back to the GEC language in that repressolator you mentioned. How does simulation help avoid a catastrophic biological failure?
It's absolutely essential. In one of the case studies, simulation revealed a defective repressolator design. The initial device, it just failed to oscillate.
Why? What went wrong?
One of the transcription factors, heresy, had a binding rate that was too strong and an unbinding rate that was way too low.
So the switch got stuck in the off position.
Exactly. It jammed. The circuit couldn't reset itself. The successful design required swapping heresy out for a different part, Lassii, which had the right dynamics. Simulation caught that before they ever built it.
And in even larger systems, like that predator-prey circuit, avoiding crosstalk must be critical.
It is. GEC modeling helped define compartments, not just for simulation, but to restrict how parts were used in the actual design. They had to make sure the same promoter, used in both the predator and the prey cells, didn't create signal interference between the 2 logic networks.
We've really spanned the entire information landscape of biology today.
We have. We started with the inherent elegance of the genetic code, a robust, hierarchical 6-bit system. Then we moved to using the physical DNA molecule as the ultimate digital archive.
And we finished by talking about how we are now imposing engineering control, using languages like LBS and GEC to design circuits, and synthesizing the actual biomolecular logic gates to perform complex operations inside living cells.
The cell, from this viewpoint, is a highly sophisticated programmable device, and the entire field of synthetic biology depends on abstracting that complexity into elegant computational rules.
So what does this all mean for you? We've seen that the genetic code perfected over billions of years is optimized for one thing: robustness, survival, minimal error. But now we have the tools, the new languages and logic gates to engineer systems for totally different objectives, for speed, for precision, for brand new functionalities. If we can define biological processes in terms of formal computer science, what new radical optimizations might we introduce into our engineered biocomputers, pushing performance far beyond the limits nature imposed on that original cell? That's something for you to mull or explore on your own.
Until the next deep dive, keep questioning the code.